Thank you, Keith. A couple of words before I introduce our expert of today. A couple of words of Bryan Garnier. So we are a pan-European independent bank. We are located in Paris, in London. We've got some offices as well in New Delhi and in New York. We are providing full-fledged services to small and mid-cap companies through different, and so we are providing corporate finance. We've got different business units. We are in corporate finance, in brokerage, and asset management. Healthcare is one of the biggest franchise of the bank, and we've done this year roughly 10 transactions in healthcare, from equity capital market to private placement or to M&A. We are about 15 professionals, senior professionals in the bank dedicated to the healthcare franchise. And within the bank, we have identified regenerative medicine as being one of the most promising area. So we are putting a lot of effort and resources behind this specific field. And so therefore, I'm absolutely pleased and honored to co-host this meeting and to moderate this panel on advanced targeted immunotherapy in cancer. And hopefully it's going to be a robust and very interesting discussion. And so before we diving into the questions, I think that's probably our different experts will introduce themselves and to provide with a little bit of background and research and interest. So, Imam, maybe you want to start?
Oh gosh, okay. Ladies first. My name's Emma Morris. I'm a physician and a scientist. I'm trained as a clinical hematologist. My background's bone marrow transplantation, so I'm one of the attending physicians in the allo transplant service at UCL, which is one of the biggest transplant programs in Europe. I have been running a T-cell immunology research program for about the last 15 years, and we've been developing gene-modified T-cells. I am currently chief investigator of 3 first-in-man phase 1 studies with gene-modified T-cells in infection and hematological malignancies. I also involved in some immune deficiency stem cell trials with gene-engineered stem cells as a principal investigator. I'm on the board of the British Society of Gene and Cell Therapy. I am a director of the BRC, the Biomedical Research Centre at UCL. I chair the GM Safety Committee for Clinical Trials across the UCL Partners Network. So I'm quite embedded in the field, I guess. We've been treating some patients. So I guess I'm immersed.
Very good.
And I've been trying to do it sort of with one man and a dog as a typical clinical academic in this space. And it's achievable.
Super. Thank you very much, Ivan.
Thank you. Thank you, JC. Thank you, JC, for the invitation, and Maury also for the invitation to be here today. My name is Jonathan Schneck. I'm a professor of medicine, pathology, and oncology at Johns Hopkins University. And I've also been in the space of T-cell biology for a very long period of time. Usually I like to start with some slides, but this is not the audience for slides. So I'd actually like everybody not to look, but to listen. Listen. You can close your minds right now and listen to an overture, an 1812 overture, or listen to a symphony. Listen to something like that. That's the way, and the reason I like that analogy is that's really what I use to explain what the immune system is about. The immune system is about a symphony. It's an orchestra that's playing, and it's a symphony that we're hearing. And when things go right, the symphony, at least to those who can appreciate it, sounds beautiful. I don't always appreciate it, but that's another problem. And as a symphony, we can identify different components of it, different parts of it. We identify T-cell arms as part, let's call it the winds, let's call it whatever part you would like. As a symphony, we identify different roles. And one of the roles in terms of a symphony is somebody to bring it together. How do you conduct that symphony? And that's really a very central role. It doesn't necessarily play an effect or function. It doesn't have a T-cell killing. But it really tells us how and when the orchestra needs to chime in. And that has always been the target of our interest. So we take it from a very basic perspective. You said you have it, you do it from a horse and a
one man and a dog. It's a very English expression. I
one man and a dog, and I'm thinking, I get half a dog. But occasionally we get to think about who is the conductor and what the conductor is. The conductor of the immune system symphony is the antigen-presenting cell, is the cell that stands at the interface between the outside world, all the threats, and tumors can be part of that outside world, and all the inside world. So it's the cell that is at the center of the focus. And that has always been our focus. How can we analyze that? And essentially what our work has been is to study, well, how do we make an artificial conductor? How do we make a pharmaceutical solution for a cell? And that's part of what we've done in terms of making an artificial antigen-presenting cell, an artificial conductor. And now we've had the great fortune of watching that. I've had the great fortune of watching that as we start commercializing it. So it's a great position that I'm in academics. I get to think about it. I get to test it, to try it in this setting or that setting in our models. But now it's very, very exciting because the whole raison d'etre, I trained clinically in pediatrics. Our whole raison d'etre of my training was ultimately to be able to see it come to man and women, come to men and children. That's part of the excitement, and part of the excitement of being here is to transfer that, and because we'll need investments for those things, and we need different structures, like Oz has talked about, in terms of how we're going to do that. We need the regulators on board. Those still remain work in progress, and it's exciting to be here at the forefront when we're going to, over the next 10, 20 years, see how this field will explode and will do good for man and women.
Super. Thank you very much. Usman.
So, thanks very much. So, Azam, Head of Cell and Gene Therapies. I'm a physician by training from the UK, originally an obstetrician and gynecologist, so my knowledge of immunology was very different from the experts that are sitting here. So I don't claim to be an immunological expert. Worked in life sciences for the past 15 years, have led and have been privileged to be parts of teams that have brought a lot of breakthrough therapies to market. So thanks for being here.
Thanks. I guess, the first question that I may have is, we heard, Karen Murphy from G, early on, and he said that, rightly, that's about, the right time to invest. But, from Novartis' viewpoint, I mean, why, to be bold, I mean, why do you think it is right now, the right time to invest in this field?
Yeah, I think going back to what I said earlier this morning, the science is ripe enough now. The clinical data is getting extremely compelling. There's been a couple of other, I think, big advancements in the past few years, and I think a big part of it is the ability now to scale up what was traditionally considered benchtop lab-based systems to something that could be industrial scale. This has really, really come of age in the past few years. So I think this is a very, very ripe time for folks to seriously consider investing, not just in the science-based companies, the biotechs, but also the enabling technology players around them. So I think this is going to be an ecosystem. So I would encourage people to think of it that way. It's not about one player, a Novartis or a big player. It's about different companies, different constructs, and very much also looking at those players that enable the ecosystem to flourish as well. And clearly there won't be investments in the academic institutes, but clearly I think creating forums whereby academics can partner in a better environment than historically we've been able to, because let's face it, there have been challenges over the years with conflict of interest and working together with academic institutions, but I think it's a realization, right, that it's getting harder for academics to get funding. I mean, my colleagues in the States in particular, struggling to get NIH grants now, even areas of oncology. there have to be other escape mechanisms for funding. And this is why I think you will see more collaborations between bigger players like Novartis with academic institutions. So I think it's the perfect time to start to consider investing in this space.
Jonathan, I know that you are involved in different biotech companies at home in the US. And so why investing now? Why the audience, the investors should really consider that, this is definitively the right time?
So I think what I want to echo a lot of Oz's view in terms of the time and the maturation. So I think what we've been doing as a model is we have the patient and science following the patient, understanding the biology of the patient, and then really directing our thoughts and our investigation into that. And so that has led to a robust development and commercialization effort. And I think that now is the right time. We know the tools. I like to say that I stand on the shoulder of giants, people who discovered these things. And so we can take engineering systems to develop what are artificial cells by simply taking reductionist systems, putting on proteins that essentially allow us to mimic things. And so it makes it more compatible in terms of and it can be used as a platform in terms of reproducibility, standardization, and scalability that now makes it the right time in terms of investment. As a platform, as a therapy, as then allowing us to make cells and to deliver cells that are now uniformly treated and uniformly cultured so that there's a standardization, what would be called SOP, standardization of process and whatnot. And so I think that that's really what drove us into it. And then ultimately I can take the academic perspective and I'll go back to the idea. That's why I love the research. I'm passionate about the research. If I have the opportunity and pleasure of meeting some of you, you can see the passion comes through right away. I can't wait to get back to lab. Been away for a few days. Can't wait to get back. But it's always been about, okay, it's nice to do what you can do in lab, but it's got to be about delivering things, like I say, real diseases, real people, real treatments. And that's to see what Novartis has been doing has been wonderful in terms of investment and that sort of structure. And I think I'd encourage you to all think along those lines and think that this is the time and the opportunity. You're getting in on the ground floor and you're going to see a lot of return on investment and good, and you'll do good in the process, which is very important.
It looks like, especially in cancer immunotherapy, the choice and the selection of the patients is one of the key elements. And we saw some impressive results around the PD-1 or PD-L1. But when it comes to the selection of the patients, how are we going to manage the selection of the patient, and especially to work with diagnostic partner? And Emma, you've been involved in different clinical trials. I mean, how to proceed?
Yeah, I think I think the key to being a good physician is actually all about deciding who are the right patients to treat. And that's one of the key skills, and most good centres will have people who are able to do that. And you need to know what your medicine is, what your drug is, what your cell therapy, what your transplant procedure is. You need to know what the underlying disease variability is within a population, and you need to know quite a bit about the fitness of the patient. And these are all things that actually are exceptionally variable, but there's nothing different about the patients we're going to be treating, or we are treating with cell and gene therapies, and patients we treat with any small molecule drug. So in the same way that other aspects of pharma are developing strategies to personalize small molecule drugs on the basis of SNP data, whole genome sequencing data, which will predict your responsiveness to an antihypertensive or whatever. The same plethora of different variables are present in our patients. Some of them are more complicated because we're dealing with a biological therapy that is immunologically active. Most of these cells will have receptors on the surface that, in order for the cell to function in the patient, the receptor has to engage with the target that's on the surface of the target cell. So, for example, a cancer cell, the chimeric antigen receptors, they will bind to a cell surface protein. The T-cell receptor therapies, which my lab is interested in, and the trials that I'm running, one of them with the support of the cell therapy catapult, uses the native T-cell receptor, so it's an alpha-beta heterodimer. It has a physiological signaling mechanism, and it interacts with the target cell through its MHC molecule. So it's more complex immunologically, it's more physiological, it's how our T cells, our killer cells, normally interact with our target cells. There are limitations with that because the T cell receptor will only bind particular types of MHC molecules. These are tissue types, what people will refer to as a tissue type. The CARs can bind any protein on the surface if it's directed to it. There isn't a limitation with tissue type. The advantage with the T-cell receptor, however, is the targets it can bind can be intracellular proteins. So there's a whole array of cancer-specific targets that will only be able to be recognized by T-cell receptors. So there are, when you're talking about T-cell therapies, there are these two approaches. The CARs have been the first to get the most substantial data out there, but they will have a very specific portfolio of things that they can target. And it may be that not everything will be as successful as targeting CD19 in B-cell leukemias. And the T-cell receptors, I think the thing we have to understand as scientists and as physicians and as investors and pharma is that every T-cell receptor or every CAR we use for a different indication and that we introduce into a different T-cell subtype will have a different function. And there will be quite a lot of variability. So it won't be like producing a drug and making a biosimilar the second time round for a slightly different indication. There will be inherently quite a lot of variability. That's probably a challenge. I wear an academic and a clinical hat. I don't wear a financial hat. There will be more risk attached, but there's potentially much more excitement. Because when something works, and if you can, some of the things we're doing in our lab is identifying motifs within the structure of a receptor that make it be, the expression of it on the cell be massively enhanced. And if you can identify those and engineer those to be in other T-cell receptor or CAR constructs, that will modify your procedures. So there's a huge amount of scientific variability. And I drifted a bit from your question, which is how you select the patient. But it's really to give you a flavour of the reality of it. Yeah. And of course, in the phase 1 trials, we are restricted to effectively using patients who have a poor prognosis, who have tried all the standard therapies, and who are predicted to be alive for long enough that you will get your endpoint data from your trials. And so a lot of the studies are using patients with older patients, which actually doesn't make for good photos. Kieran, I always think in my slightly cynical way, if you're going to do a phase 1 trial, make sure you get a really pretty looking kid that responds, because that'll look fantastic in all your presentations forevermore. You don't really want some sort of grotty looking old person with only 3 teeth, because your photos won't look so good. It's quite cynical. Anyway, Emily Whitehead is a beautiful child, and it's fantastic she's done so well, but she's a great poster girl for... I think the reality of it is people with cancer, most people with cancer are older. Mm-hmm. And we have to be able to scale these up and make this deliverable in a setting where older people can receive this therapy safely, and they can receive it ideally as an outpatient-based therapy in a clinic, and that they don't have to travel 100 miles or, if you're in the US, 500 miles to get to a centre of excellence. This is what we need to be thinking about now. How in 10 years' time can we deliver these therapies to a vast number of patients who will potentially benefit from them? Because the thing that's so brilliant about them is that a one-off infusion of cells, they'll persist forever, like those cells that responded to chickenpox when you were 3. You're not going to get a reactivation of that virus again because you've got a population of those T cells that will stay in you forever. And if we can do that with cancer-specific T cells, you don't keep having to come back and having cycles of chemotherapy. Sorry, I'm quite passionate about it too. I do think it's really exciting, but we have to think about all the challenges.
Okay. So, Usman, is that CAR against TCR, or is that CAR with TCR in terms of, especially you?
Yeah, I think, Emmett nailed it. I think they're going to be complementary therapies, right? That's the way you've got to approach this business. And I think if you look at something like, so cancer is a heterogeneous condition, okay? Even within a particular cancer, the variability can be huge. This is why I think folks in development in this space are quite judicious about selecting the right patient populations to start with. So, for example, we started with acute lymphoblastic leukemia, both pediatric and adults. So we have data in both. It's a very expressive tumor in the vast majority with CD19, but you also get non-responders as well. So, and then you start to look at the older population, CLL, diffuse large B-cell lymphoma, which are our other programs. There is more heterogeneity there. You're going to have an armamentarium of therapies available, small molecules and biologics, okay? I think this is going to be a field that's going to be, personally, I think more autologous focused to start with. Will establish in the next couple of years, soon I hope, as you can see from some of our public disclosures. But I think Emma's thoughts around how eventually this field could flourish is actually where we all want to get to. That there's a huge unmet need. You start to go up the chain in terms of other cancer types and earlier interventions, but that will take time to get there. But I think it's a realistic dream to achieve. And if you look at sort of the blood cord world, I mean, we're already there to a point, right, in terms of being able to service large geographies and large territories. But I think that's a vision that we could all get to. But it's going to take some bold maneuvers in the next couple of years, which is what a lot of people are trying to do right now.
I would support that. We mustn't forget that we have a global network that's exceptionally successful at delivering, processing, and infusing viable cell products. We do it all the time. I've been a stem cell transplant physician, consultant for 15 years. I get cells that arrive from Australia, from the US, from all over the world the whole time. It works. What we're trying to do is just genetically engineer the cells beforehand. But that's it. And so actually it's not, the infrastructure is there. It needs to be bigger, and we need to tag in the groovy science.
Exactly.
Okay, so it looks like, we've got CAR, the TCR. What about the other approaches, and what are the new technology being pursue that will hopefully deliver the similar results?
So thank you, Jesse. That is that is also one of the very, very exciting things. As people have appreciated the impact of immunotherapy, both the CARs, the TCR, what are called the checkpoint inhibitors, we're really beginning to realize the full power of the immune system. I think, well, let me step it back. I think we appreciated the full power of the immune system years and decades ago, but we're actually being able to realize how we can now deliver that. And the exquisite sensitivity that the immune system provides. Emmett gave the example that she works in a field where the T-cell receptor therapies are exquisitely sensitive and very much can be tailored designed. And we gave a little bit of analogy. I like in the US to give a little more analogy on that one. So the targets that the T-cell sees is something we call the histocompatibility molecules, or MHC molecules. MHC molecules, if you imagine my two fists as hot dog buns, in the hot dog bun, there are different hot dogs. And the T-cell receptor can differentiate between, say, an Oscar Mayer Wiener hot dog, and I don't know a favorite one here in England, but that's what the T-cell can see. So it's really... What?
Soggy cheese sandwich.
Oh, I don't know how that analogy will work. Seems to go okay in the U.S. But
Yeah,
so it's that exquisite sensitivity that's going to allow us to tailor this for individuals. Now that should have a fitness benefit to those in terms of efficacy to those individuals. So in terms of platforms and in terms of new technologies, part of it is going to be able very much to personalize that. And I think what we're about to see is the if we do it right, if we can do it right, if we can get the investments and get the systems up and running, is to see the convergence of two fields. The convergence, let's say, if I take cancer as a model, but we can think about it more broadly, the genetics of cancer, where every patient will come in and be sequenced. This is just by way of example. We'll have every patient come in and be sequenced. And in their tumor, we're going to find, we know already that for different tumors, there are different numbers of mutations. Everyone has mutations. They have all proteins that have been modified by the tumor. That's what the tumor does. It makes mutations. It escapes the regulation. But these mutations, and everybody's tumor is different. Jane's is different than Emily's, and those are all for the immune system, because the immune system, my immune system knows who I am and knows what my proteins are supposed to be like. But these mutations in the tumors now are unique. And from the immune system perspective, it's an unbelievable target. So it's something like we haven't been able to target in the past, but we can target in the future, these mutations. That should help both in terms of our TPPs, our targeted patient populations. That should help in terms of the efficacy, if we can deliver on that. And those are all going to be based then on platforms that are going to help us understand how to do that diagnostically and therapeutically. And one of the things, again, that I think is very exciting, because I can have a foot in both of those domains, is to be able to design systems. And we really have had some very breakthrough experiments recently where we can design systems where we can tell you if somebody comes in, Jane comes in with her tumor, and we have her tumor sequenced, we can make certain predictions about what might be recognized by her T cells. But actually some of the stuff that's come that the people in the lab are calling me about even here is not only can we predict them, but now we can tell you, JC, which of those are real, which of those should be targeted. And that as a platform should serve everybody well. Yeah. It's not going to be a one-man story or anything.
JC, if I could just add to that, I think the other piece that will probably revolutionize this space sooner than we think is going to be gene editing platforms. Okay. So the next wave of and this is where a lot of the biotechs are focusing right now, and appropriately so, is looking at technologies that specifically allow genome editing, building on some of the thoughts here, can be applicable to T-cell receptors, CAR therapies, non-T-cell as well. And the kind of things you're looking at are so-called zinc finger nucleases, CRISPR, TALENs technologies. These are the things that I think are also going to be, it's again going back to this enablers to allow certain platforms to be improved upon and flourish. I think that's another very explosive and exciting area of science. But I think it's also going to be the next wave of innovation in the cell and gene therapy space as well.
So the first wave of studies have all been about redefining the specificity of the T cells, so targeting it to a particular cell type. What we're all doing in our research labs, and which I'm sure many of you are aware of and are in that space, is actually trying to just alter the functional capacity or the homing ability or the persistence or the differentiation status of the cell itself. So it's not just about targeting it, instead of it recognizing A, making it recognize B. It's about actually what else can you do to that cell to enhance its performance. And that requires a lot of gene editing approaches. So, and that will be translatable to many other different diseases.
Right. Which leads to the question in terms of the not only the business model, but the partnerships, and what kind of stage of partnership is the best for a Novartis of this world. I mean, when you do some partnership with some biotech, especially in this area, would you consider early stage? Would you prefer more late stage? What's your view on that?
So our philosophy is that we're very agnostic to the timing of the partnership. Well, a couple of things that matter to us. I mean, clearly the first and most important thing is that the science, the fundamentals. Is it robust? Is it reproducible? Is the data qualifiable? We look to see clearly clinical data always helps. So if there's a maturity of inpatient data, that really is very, very helpful. I mean, the University of Penn is an example of that. It was small numbers, but the data was very well documented and very, very compelling. Earlier, I think from a business perspective is a good thing. But I think the thing that trumps it all is the chemistry of the parties at the end of the day. If you take a collaboration like Penn, there was clearly a very strong academic chemistry. If I look at my colleagues in Novartis Institute of Biomedical Research, colleagues like Bill Sellers, who's very, very renowned oncology researcher, having that connectivity in chemistry and being able to think in sync with professors like Carl June and Bruce Levine, that's also very, very important. So you need to have that, I think, that academic credibility in-house and those champions. And that speaks to culture, right? The cultural element has to be there. I think that trumps everything else. If that isn't there, then you can have the best term sheets and the best deals there. It's not going to work. So timing-wise, we're agnostic, but we look very, very importantly at the culture, the data centricity point of view, and are we going to be able to grow and have a common vision together? That's very important for us.
All right. Jonathan, did you have any view on this specific point?
Oh, I agree in terms of the culture. I'll also take the flip side, which we discussed this morning. So the cultural barriers are really substantial. And the culture at the universities also has to change and is changing, and we see that. I'm sure you've seen that as well. But it's that sort of cultural changes have to come both from the industrial side as well as the academic side. The concept of the tower, of an academic tower that's removed, which is, Johns Hopkins is thankfully pretty well entrenched in that view, or had been for quite some time. But that culture is changing and needs to change. And it needs to change from grassroots, from inside as well as from outside, in terms of people coming in and trying to support your institution, support your alma mater, support other institutions that have that vision, that can share the vision in terms of the timing. And I would say in terms of when, the earlier the better, because in the institutional, from our perspective, we can shift very quickly from in terms of we don't have heavy set infrastructures to run some of these. Obviously, some of the clinical trials are going to be different, but we have plasticity and ability to shift. And so, if we can work in parallel, as opposed to, if we can work together, as opposed to kind of going our own ways, then ultimately we'll get to places where, what were you saying, 5 years to an IND filing? I mean, an unbelievable, that's an unbelievable success. And I think that's what we want, and we want the regulators then to also understand that, okay, the culture and the mindset has changed, and so now their mindset's going to change as well. Right. Especially in these very highly personalized fields which is what this is going to be about, to be able to understand that the nature of the clinical trials and the nature of the trials are going to be different.
Right. Now, the business model of this new therapeutic approach is different. It's not the traditional classic one from small molecule that you are delivering to the patients through their GPs or specialists. So, Usman, you said earlier on that there was this issue of delivering the service at the point of care. You are absolutely right. But now, how to convince now to the investor mindset, which could be more traditional, small bid versus this kind of new delivery, new approach of treating patients.
Sure.
Because this could be a barrier
Sure.
for the investors.
Yeah, but I'd give sort of one of the good analogies, and Emmett, you touched on this a bit. If you take the bone marrow transplant world, okay, go back 20 years. I mean, doing bone marrow transplant 20 years ago, the systems were pretty immature, right? But you look at how that is a well-embedded system. There are surrogates there of service-type provisions within systems of healthcare that exist now, which can be built upon as a framework. So this isn't a new concept in that perspective, okay? Clearly there's differences when you look at geographies and approaches to that kind of model. But you're looking at feeders now coming from all over the world, as Emma said, in terms of stem cells arriving from bone marrow institutes, etc. So I think there's some precedence there. I think also you have to have a very compelling value story here for the system of healthcare, okay? So clearly you're dealing, initially if you take something like a CAR platform, we're dealing with refractory relapsing patients. The patients that have gone through, tried other therapies, and the cost investment for those patients is going to be significant. Getting through bone marrow transplantation, some of them may not be eligible for that. They may not do well with bone marrow transplantation. So I think the value story will be there. I think it will take a lot of innovative thinking in how you plug the back-end systems of a healthcare system, and I'll take a US system of healthcare example as a service model. You're going to be thinking about how do you populate and build a service of care that goes in, a team that cooperates seamlessly with that system of healthcare from a patient perspective, physician perspective, nurse practitioner perspective. That's the kind of way I think you have to look at this. And there are ways to do this. It is going to require investment, and companies like Novartis are investing. I think there'll be others that will want to enable and help us. If you think about the IT support, you think about supply chain, logistics, there are players who want to work with companies like ours in this space. So there's no shortage of appetite, and I would have to believe that all these players, including ourselves, clearly see a benefit from a business in the long term. Otherwise, we would not be doing this. Right. Clearly, we see the huge unmet need and the way we think we can transform the practice of medicine. But we also see a very viable business model. So there are fundamentals that I think exist in certain healthcare systems. And other systems, I think we've got a bit of a greenfield approach as well. If you look at other parts of the world, Pan-Asia, for example, where you're going to have other markets and other dynamics, it could be a different model. Right. But I think certainly for the mature established markets, you're looking at very much the service kind model, service type of model. It's not going to be the traditional pharma, push a pill, push a biologic and infusion center. And I think we'll start there. But I think Emma's vision shouldn't be lost that eventually as the science matures, you can see this becoming a more mainstream therapy in future years. Allogenic platforms, other things. We're not there yet. I think the science needs a lot of evolution. I think panelists would agree there that the science isn't mature yet, the clinical data isn't there yet, but it can get there over the next couple of years. So those are my thoughts on how. I don't think it's going to be, it's going to be an investment. It may be a stretch right now for folks to see it, but I think the investments and willingness to do the investments is there. I think it's a good time for folks to really start to think about this ecosystem and how there could be investment opportunities at different interfaces of that ecosystem.
Right. Emma, with your academic hat?
So I think you touched on this as earlier. One of the problems in academia is research funding. It always is. We always want more, and there's never enough for all of us, for all our fabulous ideas we have all the time. And one of the major issues with most funding bodies, certainly in Europe, and I I imagine also in the US, is that they're very interested in funding very basic research, mechanisms, models. They like us to make a proof of principle discovery, publish it, and then thank you very much. And there are some funding bodies, for example, in the UK, the MRC have schemes that support up to first-in-man studies. They're very, very expensive at the moment because at the moment in that first-in-human space, it's very heavily regulated. And for usually relatively small numbers of patients, the cost of delivering those sort of studies to get your early data is very expensive compared to other things. And then beyond that, actually the research councils, the charities that fund most of biomedicine across the world in academia are not interested in funding the actual, from phase 1 beyond, the big scale up, the how do we actually make this work, how do we deliver this to people, how do we turn this into a product. A lot of academics also lose a bit of interest because they're on to the next idea and they're back in their lab fiddling about with their mice or whatever. And so I think there's that is where we absolutely need this relationship between academia, clinicians, patients, pharma, biotech, agencies like Catapult, the regulators, to work really closely together. And I think this field is undoubtedly the one where it'll only work if we do work together.
Thank you. Do I have still the time for? Okay, good.
I have to go back to the mice. Excuse me.
Yeah. So I want to leave some time for the audience to ask the different questions that they may have. So yeah, go ahead.
It's Christian Glenn from Edison Investment Research. Just this week we had Dendreon filing for bankruptcy, effectively on struggling to commercialize their dendritic cell based cancer vaccine. I just wonder whether there's any comments, particularly from the panel, around some of the lessons that maybe could be learned from that, maybe even if it's just in terms of a process side of things. And also maybe then just leading into that, some compare and contrast the sort of that first generation cell-based vaccine versus the CAR Ts.
Sure. Do you want me to?
Go ahead.
So yeah, Dendreon, company we know very well because actually our Morris Plains facility was actually a former Dendreon facility that we acquired. So when we did the deal with the University of Pennsylvania, pretty much thereafter we actually acquired the manufacturing site in New Jersey that was originally one of the Provenge sites. And I have to give Dendreon a lot of credit in this field because they really had the courage to make the first bold investments in this space, okay? And I think they did a great job. I think the challenge that they had, frankly, was a vaccine immunotherapy platform that really showing incremental benefit over standard of care was always a struggle for them. And I think that's where the story stopped. I think had they been able to demonstrate a better benefit risk there, above and beyond standards of care, it would be a very, very different story for Dendreon. So I think you have to couch it in that. I think that the platform, the concepts, the courage they had was phenomenal. They have a lot of talent. In fact, a lot of that talent that was in New Jersey actually we acquired. And I tell you, those guys are very, very sharp when it comes to the cell and gene cell processing editing world. I think the other challenge there, and having spoken to former colleagues who were at Dendreon, some of them I know very well from former lives, really the approach as well was very much still from a commercial perspective using a traditional pharma vehicle. And I think if you look at the benefit risk of the platform, not giving as much comfort that it was above and beyond standard of care, and again using a traditional push kind of pharma model, you can see where the challenges were for that business. But I think they will have, I think, a legacy that's going to be, we'll look back and say, hey, you know what? They did it, they invested, they made the moves. And I think it's enabled others to do things. I mean, frankly, we were enabled by them in a way. If you think we took the Morris Plains facility, I mean, they really were very, very futuristic thinking in that, and I think the luck was not on their side. I think the science moved quickly, and so that tells you how important it is to stay ahead of the science and keep moving with it and have more adaptable manufacturing process systems. But I think their fundamentals were very, very strong. It just didn't pan out from the overall story at the end of the day, which is a sad. But I look at it as a good lesson learned, but I wouldn't view it as a failure.
I also wanted to comment about the Dendreon story, because I also see it as a very enabling approach. It obviously didn't work for their investors. It didn't work for the people who were part of the company. But it was a very bold move. It was a very bold move to take this approach to try autologous maturation of dendritic cells, these conductors of the immune system. But it also enabled others like myself to think about taking something that I had been working on, and I don't want to make it personal, I'm just using it as an example. There are many, many others. Now, it's not about what I do in my lab, only as a model, but taking the examples of trying to make artificial constructs, trying to make more standardized constructs that have better benefits to risk ratio. And hopefully enabling many, many others to now move into the field of cellular therapy.
Yeah. Yeah.
So a lot of the discussion today was around autologous cell therapy. Do the panel have any views on whether we'll ever find a sort of universal donor system for cellular immunotherapy?
Emma?
Yeah, I can say, I think yes, we will, but I guess I'm bound to say that. I have a phase 1 study actually using allogeneic T-cells, which we're engineering, and that's actually it's not as unusual as it sounds, because we're using that in the context of bone marrow transplantation, where actually a lot of recipients of transplant need cellular therapies after transplant, and to date pretty much everyone gets, apart from the CMV and adeno-selected cells, they will get just a pool of donor T-cells infused, which have a whole range of specificities and potentially a whole range of toxicities. So there will be a role for gene engineering of donor-derived T-cells in the context of bone marrow transplantation, which treats 30,000 people a year globally, so it's a reasonably big market. But in terms of having a universal cell, that's where the gene editing comes in. So knocking down the endogenous receptors using CRISPRs, using talons or zinc fingers. And there's a lot of work which is maturing actually quite nicely, still in the preclinical phase. Nothing's gone into patients yet. But the concept would be that you take T cells, you harvest them, you knock down all the endogenous receptors, and then you introduce what you want into the T cell, and they can be cryopreserved. And then they will effectively be a bit like giving a O-negative blood product to anyone who comes in the door smashed up. Yeah. Yes.
There's one more there.
This is Fungai Chidochi from the Cell Therapy Catapult. It's very clear as the discussion has progressed about that our industry is an ecosystem, and within that ecosystem, partnerships are key. I wanted to ask Usman firstly, as part of your due diligence process, you've spoken about how effective your partnership with UPenn worked. What would you need to see in that due diligence process if you're looking at partners in Europe? And also, same question to Emma, as a outside investor wanting to work with UCL, what would you need to see at that part of the due diligence process to create an effective partnership?
If you want me to start. So you asked the question around what we would like to see with non-US potential partners, and I assume you're talking about academic institutes and non-academic institutes, right? Yeah. So from the academic institutes, I think pretty much what we saw and what we got with Penn. So there has to be, you want to see focused champions, so that's very important for any relationship, right? You want to see a passion for the other side to take bold moves and invest as well, at risk. Because on this side of the equation, Novartis is investing very heavily, not just dollar amounts, but also resource talent when we have other priorities, right? So justifying that. And I think that's very important. So it goes back to the chemistry has to be there. Clearly, you need to have, there's going to be a platform, a bias, an agenda really from an academic standpoint, and rightly so, because that's their work. And they're focused on certain areas, and you want to be sure that you can exploit and expand that to their benefit as well. I think the other piece, looking outside of that then, is when you're looking at other partners, enablers, is particularly in the space where we are heavily dependent, is contract manufacturers, other third parties, is the ability to see how can we do really good due diligence on technical transfers. And that's a real hurdle for many in the industry, whether you're looking at an academic partner, whether you're looking at a third-party provider that may join forces with that academic to help facilitate and act those transactions. And also people being very honest about their capabilities from a capacity issue as well. Because the challenges in this space for now really are there are finite places in the world that can cell process and manufacture. That's a reality. Clearly, there are companies like Dendreon who made big, bold investments, and thank God they did, because it allowed others to flourish and enable and build off that. We did that by taking their facility in New Jersey. But moving forward, we're going to be looking at manufacturing in a very judicious way, which is why we're going to be looking very, very closely at how others are willing to co-invest and invest in this space. And having the right honest dialogue about the parties' capabilities, capacity, timings, their appetite for pushing the envelope with regulators and having those bold discussions, wanting to co-create more of the ecosystem. That's very important for a company like Novartis. So because we're investing heavily and we're willing to continue to do that, but we also want to see others co-investing and risk sharing as well, both from an intellectual capacity standpoint, but also from a practical standpoint. So that's kind of the philosophy we're kind of looking at right now when we're looking at different partners for enablement, for accelerating their platforms or actually helping us accelerate our platforms.
Unfortunately, we have to stop here, but I wanted to thank you again, our three experts, for this, very, very interesting discussion. So thank you again.
Thank you.
Wonderful.